Hi, during this class session, we're going to talk all about the science process skills. We're going to learn about both types as well. And so hopefully at the end of this class, you'll have a better understanding of the types of things you want to focus on whenever you're doing activities and you're trying to incorporate a little bit of science into it. All you have to do is think about one of these process skills and how you can just twist it right into something you're already doing. And then you've, boom, started a science lesson. So let's start by identifying the basic science process skills. These are the ones that we use all the time. Even in our everyday lives, we need these process skills in order to get through the day. And so there are 6 of them. The first one is observing. The second one is inferring. The third one is measuring. Fourth one is communicating. The fifth one is predicting. And the sixth one is classifying. And then we also have 6 others, which are known as integrated science process skills. And these are the types of skills that students would be doing if they were basically doing a science fair project or actually trying to devise their own experiments and understanding the process behind doing a scientific endeavor and where you're doing some type of scientific research or trying to investigate something. And so those 6 process skills are 1, controlling variables, 2, defining operationally, 3, formulating hypotheses, 4, interpreting data, 5, experimenting, and 6, formulating models. And so we're going to focus first the first part of class here on the basic process skills, and then we'll go into the second part of class on those integrated process skills. And so for the basic process skills, I'm going to clump observing and inferring together. And even though they are not the same thing, I'm putting them together because of the relationship they have with one another. That you actually use observations to make inferences. And it's a natural process that humans go through whenever they're looking at the world around them. They do make inferences right away based upon what they're sensing with their senses, which we'll talk all about in the next class session whenever you learn about Echo the Bat. And so what I want to do here is clearly delineate the difference between observations and inferences. And also I want to show the relationship between the two of them. And so when we're observing, we're using our senses such as sight, touch, taste, sound, and smelling to gather information about an event or an object, something that's taking place. It could be in an experimental activity, or it could just be outside, walking down the street, going to a store. Humans are natural observers. And so your students are going to be really good at making observations. They're also going to be pretty good at making inferences, which is an educated guess based off of sensed observations. Now, before we get too far into talking about this point, I want to talk about this quotated educated guess. Many people have probably heard as a hypothesis being an educated guess, whereas that's really not true. Okay, an inference can be an educated guess, but a hypothesis is actually based upon observable evidence relating to the topic. Like there's a little bit more research involved, there's a little bit more information available to you that helps you create the construction of the hypothesis, whereas an inference can just be very simple, an educated guess. Okay, and so but we still use observations to make inferences like we would if we were making a hypothesis. It's just that a hypothesis usually relies on a scientific observation to take place. And so in terms of observing, an example might be that this rock feels round while this rock feels jagged. Those are 2 observations about rocks and their texture in particular. And so an inference would be an educated guess based upon those observations. And so we could say that the jagged rock must have been a bigger rock that is now broken into smaller pieces. Okay, and so notice that an inference is really defining what it is whereas observing is just describing qualities or quantities about a particular object, not making a guess as to why or how. It's just stating what is right there in front of you. And so students will oftentimes will mix up these two when they're working on something, and you ask them to do observations, they're going to instinctively give you an inference. So I think this is what's happening in the picture. I think this is an image of that. And so what they're doing is they're jumping to a conclusion about something because their body is hardwired to naturally do that, to survive. Whereas it can also be a bad thing to do that in terms of science if you jump straight to an inference, because by jumping straight to an inference, you are introducing scientific bias, because you are determined that the thing you're looking at is exactly what it is without considering every single observable truth about that particular image or situation that's taking place. And so to really get good at doing science, we need to train our students to get better at making observations and all the possible observations that they can before they make an inference. So we want to be careful not to jump into making inferences about things. And remember, one of the keys to teaching the nature of science is that not only does science rely on empirical evidence, but science is also open to change with more information as it's would necessarily say discovered, but found or communicated, even just more evidence that's been acquired through the science methods. And so scientists all the time are revising their theories, and we see that in history. A lot of inferences that were made have been changed based upon new information that's been presented. Okay, on to measuring. And so measuring is whenever we either use a non-standard or a standard way to measure something, or give an estimate on a quantity relating to like the dimensions of something. And so here I have an image of a screwdriver, just the tip where I've been measuring, and I'm trying to figure out how long the screwdriver is. And notice just how by changing your perspective or the angle at which your eyes are with respect to the thing you're measuring, how this measurement can appear to change. So this is me looking at it from the right. Notice how the line here is closer to 1/8 of an inch, or 9 1/8 inches, I should say. And then here it moves up an extra 1/16. So we have 3/16 of an inch. And then if we look at it from the other side, it looks like it's only 9 1/16 of an inch. And so measuring is something that is really important to help us determine responses to things that are happening in an experiment, or what's happening in our environment, how things are responding. And it's very important to know, though, that the way that you collect your measurements is super important, because accuracy could really affect the results of an experiment. And so to make your most accurate measurements, you need to be mindful of something known as parallax. And so parallax is it's an observed change in position of an object based upon the reference point in which you view it. And so allow me just to come back on and demonstrate here a very simple way to demonstrate parallax to students. And so if you put your thumb up and hold it out away from your face like this, if you close just one eye and look at your thumb with the eye that's open, and then switch which eye is open, and if you need to do it back and forth over and over again, kind of like this, go right ahead. And what you'll see with your eyes is that it looks like your thumb is moving left and right. But if you looked at the video, when I was doing it, my thumb didn't move at all. Maybe just a little bit whenever I've been talking, but it's not moving like this, which is what you see whenever you test this out to yourself. And so just by a different light ray going in a different angle to an object to observe it, makes it appear like it's in a different position. And so when we have measurements, if we're not good about aligning our eyes directly with the thing that we're measuring, we're going to introduce error due to this phenomenon of parallax. So good measurements are good to take. And in a later video today, when we do butterfly chromatography, we'll get into the specifics of errors in measurement, as well as how you report the certainty of a measurement. And so I'll talk a little bit more now about different types of errors. You can even have systematical errors in which, like, the instrument that you're using to measure with could be calibrated incorrectly and not report an actual accurate measurement. And so you would see that error in every measurement you make, even if you're making a really good measurement and keeping parallax in mind while you're making your measurements. And so that's just a fault on the creation of the instrument or how the instrument is operated, right? So it could be human error as well. So making sure you make error-free measurements is super important. And so it's actually really hard for students to measure objects, okay? Whether they're using a scale to measure the weight of something, a ruler to measure the width or height of something. The problem is they have a difficult time, first of all, making an accurate measurement because of the phenomenon of parallax. But the second thing is they usually don't know how to read the gradations or the markings of the scale that they're using, right? And particularly with inches, this involves fractions of an inch, right? And so knowing how your unit system is divided will help you out a lot. And so one reason why I like using the metric system rather than the English system of units is that it's based off of place value, right? And so each line is like a millimeter, whereas a bigger line is a centimeter, and then a whole bigger line then is an entire decimeter, and then up to an entire meter's length. And so those are easier to count, whereas here I'm using fractions, right? I have to count the number of lines between each of these big divisions. So from 8 to 9, if I count 16 lines, that means that each little marking is a 16th. And so here I have 2 16ths, which, by the way, is reduced to 1/8. And then as I continue up to this, I get larger divisions. So I go from 1/8 to 1/4 of an inch or 1/4 inch here at this longer one, and then another eighth inch marking, or here's another 16th marking, and they add up. And so that's really difficult for students. And so giving them the practice of figuring out how to get a measurement is important, and then also making sure they're reporting measurements with the correct number of digits or the amount of certainty is something that they need practice with all the time. So anytime you're doing an activity, it'd be very easy to throw in something with measurement. So just keep that in mind. Okay, the next basic process skill is communicating. And so this is using writing, charts, and graphs to explain events. And so particularly in science, we have to use models and charts and graphs to actually report back the information that we observe during an experiment. And so being able to construct these things is a skill in itself, and also being able to write about them in a way that uses scientific terms correctly within the structure of the language that you're using to write it with is super important. And so this photo is just a particular example of how a young child could start developing their ideas about how to communicate an observable phenomenon. So they're making like a bar chart. So they've opened up the bag of M&Ms and they've sorted by color, and then he has counted the number of color of that particular color in the M&Ms bag, and have reported it here on this chart. So across the bottom axis is the color, and then there's no label here on the vertical axis, but considering this is a really small child, I believe that this photo was taken before this was added. So this is to help the student to understand the different elements of what you need to put on a graph. There's also no title. So of course, my experience and my age allows me to make a lot of inferences about what this student is communicating to me through this image. And so it's up to us to teach our students how to write and put these properties on graphs and charts and how to write about them to communicate the meaning, right? It's very easy for me to understand what his experiment is based upon this, but with more technical graphs that are created, I really would have to have a description to help explain what it is that happened and what the information means that I'm looking at. Okay, so predicting is the next basic process skill. And so using a pattern or evidence to state an outcome of the future. Now, a prediction doesn't have to be true. It's just seen as something that you think is probable based upon historical information. And so what we're looking at right now would be a graph that is composed of two different sets of data. And so the blue lines represent the amount of carbon dioxide in parts per million measured in the atmosphere based upon ice core samples and present day data. And in red is the average global temperature of our planet over those years. And so what you'll see is that they pretty much match each other, right? And so by seeing this increase in blue and then decrease in blue, followed by an increase in red, and then shortly after a decrease in the red, this indicates that the temperature change comes after the amount of carbon dioxide is already in the atmosphere, right? They're lined up across the horizontal axis here as years before present day. So this is time. And so notice the peaks of these do not quite line up on the exact year. It's after the fact that the temperature is seen to mimic the carbon dioxide concentrations in the atmosphere. But the synchronicity of this graph leads the scientists to make the prediction that the temperature here is going to spike up tremendously along with because of the carbon dioxide concentration spiking. And so you probably have heard about this at some point. This is the idea behind global warming, that our planet is warming on average due to the industrial boom and the amount of carbon dioxide emissions that take place, and add fossil fuel carbon reservoirs into carbon dioxide greenhouse gas form reservoir in the atmosphere, which helps insulate the earth, but an increased amount kind of is like leaving that blanket on for too long, and it warms up the planet too much. And so this is a prediction, and the scary thing about this prediction is this rapid change. And so figuring out if the average temperature of our planet is going to respond similarly is going to happen at some point, and it could change really rapidly and be an issue. And so also what's scary about this chart is that this is the highest the carbon dioxide concentration has been in historical record ever, right? And we've never had any of these peaks above 300 parts per million. And so we've gone a whole 100 parts per million above the previous threshold. So what does that mean for the temperature, which has only been above positive 4 degrees from this average here? Okay, and then another science process skill of classifying. And so here we can take a look at different plants. We can observe their germination patterns and count the number of cotyledons that come out. And then we will see patterns as well with that. So we will see the leaf structure change depending on the number of cotyledons, and we will see the vascular bundles within plants' vascular system different. And so, and then we'll also see the number of flowering parts on that plant be different as well. And so these generalizations, we can take all of these plants and group them into a very broad category. Even though there's many different types of plants that are in this category and can be categorized within it, the overall pattern here is that if we see a plant with all of these properties, they're a dicot, meaning that they have two cotyledons is the leading connection here to all these properties. And then if I see a plant with these properties, it's very probable that it is a monocot, and the seed will only have one cotyledon whenever it's germinating. And so we can classify plants based upon their properties, but also use those classifications to bring us to awareness of something. And so classifying things is super important because it can lead to more scientific knowledge that we maybe weren't actually experimenting with to begin with. Okay, so those are the basic process skills. Those are things that you can very simply incorporate into a lesson, whereas the integrated process skills are still as important, but these process skills are things that you're going to have to put a little bit more effort into constructing an entire lesson in order to practice them, because these process skills are the ones that are like developing a laboratory experiment. Okay, and so when we talk later this week about scientific inquiry, it's a really good idea to have already helped your students understand some of these integrated process skills, that they're going to have to use these to come up with an experiment that they're going to create whenever they're answering your question in an inquiry activity. And so the first integrated process skill we're going to talk about is one of my favorites, is the controlling variables process skill. And so with this, the student would understand how to assign a single variable to test and only a single variable within an experiment, while keeping all the other possible variables constant, unchanged. Because if we introduce too many variables within an experiment, it will be inconclusive about why the results look the way that they do. And so if we can limit it to just testing one particular variable at a time, then we learn a lot about that one particular variable, and then we can associate it to how it leads to the overall idea of all of these variables interacting together. And so, for example, you could devise a particular experiment into different groups. And so all of the samples in group A are going to be the sunflower seeds a million brand of sunflower plant that you're trying to grow an experiment. And then you're going to use a grow light to help grow the sunflower, maybe in a greenhouse or inside somewhere. And so you're going to use a red grow light. And then you're going to have 250 milliliters of water at 6 a.m. every day is maybe your watering process. And then you're going to make sure that each seed is in a round pot that's type A. It's just some generic type I'm throwing out there. Whatever, they're all identical is the point here. And then 8 inches of soil will be the depth of the soil that I put in the pot whenever I have the seed in there. And so I'll put the seeds within the same depth within the soil also. I didn't write that on the chart, but it should go there. And so in group B, you'll notice that the only thing that's different here would be the grow light color. I'm going to use blue grow light. And so that's the independent variable. That's the variable that is different from each experimental group. And it's the thing that's going to cause the difference or the changes that we see in our results. So that's the thing that we're testing, is which grow light or the different colors of grow lights are what we're testing. And then you also notice that it's important to create a control group to compare to. And so here in group C, the variable is no color grow light. And so just no particular color. I'm just going to use the natural type of light that's in the room as a comparison. And so it's important to understand how to have it, what a control would be, and how to have a control and also include it. And moving right on into the next one, it kind of links to it, is called defining operationally. And so understanding how changing that variable is going to cause a response, and what is it about that response that you want to measure to use as your evidence for the change that occurred. And so this is how you measure the results of the experiment, or also known as the dependent variable. And it's called the dependent variable because this information depends on what you select as the independent variable, which is different from all the other variables in the experiment. And so an example here from our experiment as a dependent variable would be how much the sunflower grows, and specifying how it's going to be measured. So it's going to be measured in centimeters each day for 2 weeks. And so also I didn't clearly specify here. I made a huge assumption here that folks would understand this, but I need to measure that if this is going to be the stem height, right? There are a lot of factors about the growth that I could measure. And so we could redefine this statement for our dependent variable to make it more accurate for our experiment. And helping your students develop that understanding of specifically designing the dependent variable is super important. And that's why this is the defining operational process skill. Okay, and then the third process skill for the integrated section would be formulating a hypothesis. And so it is important that you formulate a hypothesis before you conduct the experiment, but too many times what I've seen whenever I'm judging science fair projects or just observing students is that they will try to make their hypothesis before they really develop the procedure and do the research. And so you don't want to actually perform the procedure and get the results, but you do want to have enough understanding of the definition of what you're doing to write a really good hypothesis. Too many times students will write the hypothesis and then they will make an experiment that doesn't match the hypothesis that they're making. And so that hypothesis needs to be a statement that's testable. And that's what makes a hypothesis. And so if it's not something you're testing in the actual experiment you come up with, there's going to be no correlation here between your conclusion and the hypothesis that you've written. And so my preference is think about and plan the procedure, the factors that I'm testing, outline it, and do a little bit of research on the variable that I'm testing. So for example, in this experiment, we were testing the blue grow light and the red grow light. And so what makes the blue and the red different from one another besides the color? What scientific knowledge is out there about red lights and blue lights that I can use to help me think about my hypothesis? And so I could do a little bit of research on that and realize, well, that these different colors of light indicate different energies and different frequencies and wavelengths of light rays. And so from that knowledge, which I would include in a background section, I could write my hypothesis. And so I would have learned that blue energy or blue light has more energy and that red light has less energy associated with it. And so without knowing the results now and just knowing that research, I can make a statement about what I think is going to happen in my experiment. And so here's an example hypothesis. The light from the blue grow light has more energy. So the sunflower growing under the blue grow light will grow the fastest. And so this is kind of stated a little bit as a conclusion. I like for them to be if-then statements instead. And so I would really prefer revising this to say, if the energy associated with blue light coming from the blue grow light is higher, then, so comma, then the sunflowers growing under the influence of the blue grow light, well, should have, I like should instead of will. It sounds a little bit more truthful about the way you're going about the experiment. Should have a higher stem height within 2 weeks, right? And so just by me showing you that shows you how important it is to help your students revise those hypotheses as well. Because I already want to revise mine, and I've been doing hypothesis writing for years now. Okay, and then the next integrated process skill, which is really important, is being able to interpret data. And so this is organizing your results. So after you perform the experiment, you have photo evidence of maybe the heights of plants. You've recorded measurements of them by day, and you have it kept in a log. And it could be in a really like raw format, what we call raw data. And then you're going to neaten that up by turning it into a presentable chart, table, or a graph. And that will help you in communicating the results of your experiment. And so they go hand in hand with the communicating part. And so that's why we need to interpret what it is that we got. And so here I have a graph, speed versus time graph. We've got speed in meters per second on the vertical axis, time in seconds on the horizontal axis, and they're clearly labeled and they're scaled appropriately to give us a good looking graph. And so here, the speed, I've made up a scenario to kind of interpret the data. So this isn't really an experiment that was done, but this is just an example. And so if the experiment that was done was the skateboarder, we were measuring his speed at different time intervals when he was taking off. The speed of the skateboarder here increased throughout the entire trial, which indicates a positive acceleration. And so that's our first statement. That's our first conclusion. And then you can build onto this. However, the speed does not change at a constant rate. That's the second conclusion. And then here's the explanation of that interpretation to help readers understand why you're making that claim. And so the speed was observed as 3 meters per second at the first second, 7 meters per second on the second, 12 meters per second the third, and then 18 meters per second the next. And then it continues on. It's a nice sequence here. It's still going to explain this first statement that we've made. A skateboarder with constant acceleration of 3 meters per second would change from 0 meters per second to 3 meters per second in the first second. During the next second, the speed would only reach 6 meters per second, followed by 12 meters per second on the third. Finally, the fourth second would have been a speed of only 15 meters per second. And so this is showing a contradiction between what it means to be constantly accelerating and what the results of the experiment are indicating with the measurements that were taken. And so having measurements is the first step in being able to interpret the data also. If you don't have good measurements, you can't interpret the data. And you'll also notice that having that background information, just like in constructing the hypothesis, you need to have some of it in order to write a really good description to explain your interpretation as well. And so the final statement here is that the skateboarder's acceleration was positive, but it was variable. Okay, on to experimenting. And so this one might be a little bit more obvious, and I won't spend too much time on it, but experimenting is really the act of accurately performing an experiment. So it's a combination of all of these other processes I've talked about. Being able to do research well, being able to formulate a testable statement in what we call a hypothesis. Excuse me. Being able to form a hypothesis. And a hypothesis that goes along with an experimental procedure that I've set up where I've controlled the variables to get an accurate and conclusive set of data, and then interpreting that data into a conclusion. It's basically the scientific method is really what experimenting is. So remember, not all science is done with an experiment and setting up an experiment. Some of the things actually happen out of order, and then eventually you'll have enough of this to start generating enough research points and have enough observations to actually be able to devise an experiment that you could actually test a variable to get even more evidence, or what we would call experimental evidence rather than observational or historical evidence, right? And so experimenting really is the act of participating in the scientific method. Okay, and then the next one, and I do believe is the last one of the integrated process skills, is formulating models. And so formulating models are super important in communicating. Again, another communication here. It's not just graphs and charts, but it's being able to show with a model, a process, with all of the components of the process identified and how they behave during that process, right? And having evidence to support your model is super important. And being able to, in writing, relate the evidence to a picture format is a skill that students need to develop. And so here we've got a model of guard cells, which are on the underside of plant leaves. And so these are known as the stoma or stomata. The difference is there is stomata or stomates is the plural form. And so what you'll see here on the left is the model describing that the stoma opens, a stoma opens whenever the guard cells are swollen. And so it's not written here, but something that we could do practicing our writing in science is explaining what's happening in a model. And so an explanation that I see here is that there is plenty of water, which are the red dots, inside of what's identified as a vacuole, which is the plant's kind of waste chamber in the cell, or a cell's kind of waste chamber. And there are also, or sorry, the water is in blue. The red dots are K+ ions or potassium ions. And so we can see that the water is mixed with a lot more potassium ions in the vacuole of a swollen guard cell. Whereas the potassium leaves and comes out of the vacuole and leaves the water behind in the vacuole whenever the guard cells need to shrink. And so it's this exchange of potassium ions from the cells into cells that are surrounding these guard cells that actually manipulate the opening and closing of guard cells. And that is what allows carbon dioxide to enter the plant from the bottom of the leaves so that photosynthesis can take place in the chloroplasts, or in the cells, where the chloroplasts bring in the light and the chemical reaction can take place within. And so also water can leave the plant cell through this opening as well. And so being a plant being able to balance the amount of CO2 and water that passes through the openings of these stoma is really important. And so this model does a really good job of describing pictorially what are the components of the plant cells that are involved in this process and how those components change from one phase to the next, which is a good thing for visual in every model. And of course, we could have some writing to back it up. Okay, so that brings us to the end of the science process skills. Remember, in this video, we talked about basic science process skills, and then we also talked about the integrated process skills, which are listed on the first two slides. So whenever you're working on your journal, you can go back to those two slides and refer to those as a list of something to talk about when you're reflecting. And so in your reflection for this class, I want you to, number one, the first item here is describe your experience level with implementing lessons containing instruction on science process skills, or just maybe doing a science process skill without instruction as well. And then not really question, but item number two is describe how you explicitly teach those science process skills to students. Do you teach skills, for example, in like a unit at the beginning of the semester? And Or do you just embed your process skills rather than directly teaching students about them? You just kind of naturally put them into activities. And then the last item, number 3, what science process skills do you find hard to teach and maybe sometimes omit? And why does that happen, in your opinion, in your reflection? And so also, I'm really looking forward to hearing about how you can do a better job with this skill. So in your reflection, what do you think you can do within the 4 walls of your classroom with the activities that you already do on a regular basis? How can you improve those to help you do a better job of teaching the science process skills, both basic and integrated ones? Okay. As always, if you guys have questions, email me or get in touch with me through the Google Classroom. Hope you have a great day.